My name is Forrest Craig from WF West High School in Chehalis, Washington, and my lab research experiment is determining the effect of plant wounding on the transformation efficiency of Arabidopsis thaliana. What benefits may come from genetically modifying plants? There are many advantages that can occur from this procedure, one of which being the integration of beneficial genes that are non-existent in a pre-existing plant species. Others among this include an increase in productivity and plant health, as well as a lengthened lifespan and immunity to disease. There are several ways that the genetic modification of plant life can be obtained. One method, microparticle bombardment, involves an electronic process of forcing a transformed bacteria within the plant cells. Other methods include tissue cell culture, vacuum infiltration, and what was used in this experiment, the floral dip method. The floral dip method involves an infiltration media that has the presence of agrobacterium, a natural mode of genetic modification in which the bacteria integrate some of its own DNA into the plant cells. Plants are generally dipped in this media, however, for the purpose of this experiment, the media will be sprayed onto the plants. Just as there are multiple ways of infecting plants, there are several different ways of transforming agrobacterium as well. Among the most popular are the freeze-thaw method, which is like a reverse heat shock, triparental mating, and by far the most common and efficient, electroporation. The process of electroporation is dependent upon electrical pulses of varying intensity that allows the passage of DNA past the cell membrane. The purpose of this experiment is to test whether or not wounding the Arabidopsis thaliana plant will have an effect on the final transformation rate as opposed to infiltrating the plant in its healthiest state. It is hypothesized that because the process of plant wounding under immature and unopened flower buds would provide a clearer, faster route within the plant, the process of wounding Arabidopsis plants before infection will indeed increase transformation efficiency. This will be done by inserting the GFP gene into the plants and tested by counting how many plants visibly glow in the dark. These are the methods and procedures that were used along the process of this experiment. First step to starting this experiment was appropriately getting the plants growing. 54 4-inch planters were washed, 18 each were placed in one of the 3 planting trays. Seeds were then placed in a beaker of water and pipetted and dispersed evenly in each tray. Approximately 200 seeds were planted in each tray. For the preparation of the agrobacterium, 2 LB agar dishes were made, 25 microliters of agrobacterium spread per plate and incubated overnight. From the plate growth, an OSC was made, and from that a midlog. The cells of the midlog were spun down and washed, and afterwards were placed in 40 microliter aliquots. Once the cells were prepared, they were ready for electroporation. The neon transfection system was prepared by filling an electroporation tube with 3 milliliters of buffer E2. 2 reactions were prepared by loading each of the 2 epitubes with 15 micrograms of the binary vector P-glow and the equivalent of 2 times 10 to the 6th, or 2 million agrobacterium cells. Then each epi was brought to a total volume of 100 microliters using resuspension buffer R, sucked up using the neon pipette, and docked in the neon pipette station. The electroporator was optimized for 7,000 volts with a width of 50 ms and pulsed 5 times. Once the reactions were done, each 100 microliter reaction was used to inoculate 2 pre-warmed cultures of LB broth. For the preparation of the infiltration media, these cultures were used to grow the cells on 2 more LB agar plates. The plates were prepared the same as before, however had kanamycin added to them to kill any remaining P-glow plasmids. From this cell growth, an OSC was made, and from that a midlog, from which the cells were harvested. The infiltration media was prepared by dissolving 50 grams of sucrose, 0.04% total volume Sil-Wet L77, and the harvested cells in 1 liter of sterile water. Once the infiltration media was created, the plants were treated. The first tray received no media as a control, the second tray was simply sprayed with the media until the plants were visibly dripping, and the third tray, whose immature buds were removed 2 days earlier, were sprayed likewise. The final step of this experiment was to collect the seeds from all 3 trays and plant the offspring generation as the parent generation was before. Test cases will be screened for success once the offspring generation has time to grow and start visibly glowing in the dark. However, no results have been obtained yet, as the experiment is still in progress. For assistance during the experimental process, I would like to thank Shelby Weeks, a WF West graduate whose information on a similar experiment was vital, my teacher, Mr. Henry Weeks, his sister, Wendy Neal, who ran our class in his absence, and Nancy Hutchison of Fred Hutchinson Cancer Research Center for providing me with the book Agrobacterium Protocols Volume 2 by Can Wang, from which many of the procedures of this experiment were used from.